Okay, so this is the second part of Learning Unit 3 covering the cardiovascular and respiratory systems. This actually went with the material from last week on respiratory physiology, and I'm really sorry for the delay in posting it. Remember that this is covering, the heart, blood vessels and circulation, the respiratory system. In lab, you had your first lab exam on Friday, but this coming Friday, you'll be working on the cardiac cycle and blood pressure. So hopefully kind of that delay gives you time to catch up on the material from lecture and start practicing it when you get into lab. These are the learning objectives from that first part of the lecture. So about the cardiovascular system. Our learning objectives for the second part on respiratory physiology are listed here. Again, like last time, I'm not going to take the time to read through them all individually. They're highlighted on the what should you focus on portion of the module, as well as in the section header. I did want to do a quick review of anatomy, nothing in depth, but just in case it's been a while since you took your anatomy class, focusing on the respiratory system. So when we're looking at the respiratory system, there's a couple of different anatomical and physiological zones. There's the conducting zone, which are the organs and structures not directly involved in gas exchange. These are things like the nasal and oral cavities, the pharynx, the larynx, and the trachea, as well as the top of the bronchi. There's also the respiratory zone, which are the specific structures involved in gas exchange, so the alveoli and the respiratory bronchioles. So again, the conducting zone, the nose and adjacent structures, pharynx, larynx, trachea, and bronchial tree. The respiratory zone, those respiratory bronchioles, and specifically the alveoli. Remember that alveoli are arranged into these alveolar sacs, where you have these kind of sacs of air that are completely surrounded by capillaries. So one thing to note when we talk about the pulmonary capillaries in this lecture, we're talking about these capillary beds that are wrapped around the alveoli. In terms of the gross anatomy of the lungs, the right lung has 3 lobes, the left lung has 2 lobes, and they have very high surface area. So remember, this is an important site for gas exchange. So this lung tissue has very high surface area, around 70 meters squared, and that's in those alveolar sacs. The reason that the left lung has only 2 lobes instead of 3 is because the heart fits into that spot. It's on the left side of your body, and that little gap in the lung where the heart sits is called the cardiac notch. Another thing to note is that the lungs are surrounded by membranes called the pleurae. There's the visceral pleura, which are immediately superficial to the lungs. Remember, visceral indicates that it's something to do with organs. So these are the membranes that are directly surrounding the lungs. The parietal pleurae are more superficial. They connect to the thoracic wall and the diaphragm. One thing to pay attention to is this pleural cavity, because this gap between the parietal pleura and the visceral pleura and that pleural cavity is going to come into play when we're talking about different types of pressure. Okay, so now let's get into respiratory physiology. First, we're going to go through section 22.2, talking about the process of breathing. You should focus on pressure differences that drive pulmonary ventilation, just a fancy way of saying breathing. We'll start looking at spirograms, looking at lung volume and capacity, and thinking about ways that breathing is regulated. Then we'll get into section 22.3 on gas exchange, thinking about partial pressure, visualizing partial pressure change, distinguishing internal and external respiration, and going over that decompression sickness case study from week 1. We'll get into section 22.4 about the transport of gases, so thinking about how oxygen and carbon dioxide are transported and bound differently in the bloodstream. And then our last section, 22.5, modifications in respiratory function, just briefly goes over some different conditions and how they affect respiratory gases and blood chemistry, as well as kind of the opposite of decompression sickness, which is altitude sickness. So starting with that first section, thinking about pressure differences. So in terms of what pulmonary ventilation actually is, breathing, it fundamentally comes down to the movement of gas and gas exchange. So it turns out the pressure differences between the atmosphere, so that P atmosphere, ATM, the alveoli in the lungs, PALV, and the pleural cavity, PIP, are what's actually driving that breathing. So pressure differences in cavities as well as partial pressure differences of individual gases are going to be super crucial for today. We describe kind of the relationship between the pressure and the cavity that's kind of doing this movement using Boyle's law. So Boyle's law describes the relationship between volume and pressure when you're keeping temperature constant. So you might have seen this formula before, P1V1 equals P2V2. And what that's telling you is that as volume increases, pressure is going to decrease accordingly. As volume decreases, pressure is going to increase. So we say that volume and pressure are inversely proportional. And you can see that diagram on the right, kind of demonstrating this. If the concentration of the gas is consistent and you're decreasing the space available, that gas is moving around a lot, or maybe at the same pace, but it ends up hitting the walls a lot more. That means that the pressure is higher. So constraining that space available means that the pressure is increased. So we see those differences in pressure on this diagram here. You can see things like transpulmonary pressure, intrapleural pressure, alveolar pressure, and that 760 millimeters of mercury, that's what that mmHg means. That's going to be an important number that we'll talk about consistently. So when we're thinking about how gas moves, just like in diffusion, a lot of this really comes down to going from high to low. So gases move from areas of high pressure to areas of low pressure. They move along a pressure gradient. That's what this GIF is showing. It's showing the pressure of the thoracic cavity, and you can kind of see it fluctuating as this person is breathing. One thing to note is that you can see the diaphragm moving. That's that big sheet of dark muscle, and so that's going to be important in a little bit. If you zoom in, you can see the alveoli with those pulmonary capillaries surrounding them, and you see oxygen and CO2 moving. So that 760 millimeters of mercury is actually the atmospheric pressure that's being forced on our body by gases in the air at sea level. So kind of keeping things consistent, not going to high altitudes, not going to the depths of the ocean. This is the force that atmospheric gases exert on our body. So 760 millimeters of mercury. And usually when we're looking at different gases within the thoracic cavity, we kind of compare them to that 760 millimeters of mercury number of atmospheric pressure. So if you look in this image, you can see that the intraalveolar pressure, for example, is listed as 760 millimeters of mercury, but then in parentheses it says 0. And the reason for that is because there's no difference between that alveolar pressure and the pressure of the atmosphere. For intrapleural pressure, that's 756 millimeters of mercury, and so that's 4 less than the atmospheric pressure. So here we have -4 millimeters of mercury listed for intrapleural pressure. So when we're looking at intraalveolar pressure and intrapleural pressure, we shorten those to P.lv and PIP. Another word for intraalveolar pressure is intrapulmonary pressure. Just to unpack that really briefly, so intra means inside of, alveolar means the alveoli, pulmonary means lung. So when we're talking about pressure differences, alveolar is basically the more technical term for pulmonary because we're talking about what's going on inside of the lungs. So again, intraalveolar is the same as saying intrapulmonary. So as you breathe, as you go through different phases of breathing, the intraalveolar pressure actually fluctuates But then the thing to remember is that we have this conducting system where our trachea and our bronchioles are connected directly to the outside environment. Our alveoli are connected to the atmosphere as well. And so because everything's connected, even though it's changing, it eventually equalizes with that atmospheric pressure. The intrapleural pressure, on the other hand, PIP, changes through those different phases, but is actually always lower than atmospheric pressure. It's negative, that's affected by lung elasticity, by alveolar fluid, by pleural fluid, and by thoracic wall elasticity, but it's always lower than that intraalveolar pressure and that atmospheric pressure. And transpulmonary pressure is defined as the difference between that alveolar pressure and that intrapleural pressure. And I think those figures should actually be reversed. I think it's P.lv minus PIP. Sorry for the typo. I'll try to fix that. But it's not something that you need to know for the exam. Just know that transpulmonary is the difference between intrapleural and intraalveolar. So I showed you on that GIF that as that pressure changes, we see the diaphragm contracting and relaxing. So there's a couple different muscle groups that are involved in changing the volume of the thoracic cavity, and in doing so, changing the pressure. So this is one of those physical factors that affect ventilation. On the left, we see the diaphragm highlighted in green. It's that big sheet of muscle that separates your thoracic and abdominal cavities. On the right, we see the intercostal muscles that are between your ribs. So those are in your rib cage, the bulk of your thoracic cavity. And so as those contract, they change the volume of the thoracic cavity. Increasing volume decreases pressure, and vice versa. And so it's not just the muscles, it's also kind of the tissue of the thoracic wall, the elasticity of it. So all of that kind of ties together, thinking about how this thoracic cavity moves. A couple of other factors that affect ventilation include characteristics of the lung tissue. So when we're thinking about airways specifically, we call that tubular diameter. If the diameter is smaller, there's a lot more resistance. And vice versa, if the diameter of those airways is larger, there's lower resistance. There's also alveolar fluid. So in this image on the right, you can see that there's a couple of different cell types within the alveolus. One of those is type 2. It's a surfactant secreting cell. And so this fluid is very important because the alveoli are so tiny that they would collapse if they didn't have this surfactant. So the surfactant is very important for managing surface tension and preventing collapse of the alveoli. Okay, so kind of putting those ideas together and thinking about this pressure-volume relationship, when you go through inhalation, that's also called inspiration. Those are just two different terms for inhaling. So what's happening there on a muscle level is that the diaphragm and the intercostal muscles are contracting. That's increasing the volume of the lungs. So they contract and the diaphragm kind of pulls down. The rib cage pulls out, and so that's going to decrease the pressure of the thoracic cavity and draw air into the lungs. And if you take a deep breath just for a moment, you can really feel that. You can feel your diaphragm dropping. You can feel your rib cage stretching out as your intercostal muscles contract. So that's increasing that volume and decreasing pressure of the thoracic cavity. Remember that the pressure is going to go from where the air is going to go from where there's high pressure to low pressure. And so at this point, the outside environment has higher pressure, the inside of your airways has lower pressure, your lungs have lower pressure, and so that's going to draw that air from the atmosphere into your lungs. For exhalation, which is also called expiration, those muscles just relax, so then your diaphragm goes back up, your intercostal muscles kind of come back together. That decreases the volume of the thoracic cavity and increases pressure, so then air is going to flow out from your lungs into the atmosphere. So it's going to go from this higher pressure environment inside of your lungs, back out the airways, and then into the atmosphere. So just think about that kind of contraction and relaxation. The relaxing part of it is a passive process. So this doesn't require extra work. It's just those muscles relaxing again and kind of reverting back to their natural state. So when we go through inhalation and exhalation, that equates to one respiratory cycle, one sequence of inhalation and exhalation. And we can see that in this here. Actually, not yet. Sorry, I thought we were a couple slides in advance. So when we're thinking about that kind of process of inhaling and exhaling, of the diaphragm and intercostal muscles moving, there's a couple of different types of breathing that we are kind of thinking of. So one is eunia. This is just quiet breathing. There's no cognitive thought involved. It's kind of that weird thing where like you're just kind of breathing and looking at your phone, and then you see one of those memes that are like, and now you're manually controlling your breath, because we just kind of don't have any cognitive requirement for breathing when we're in this state. And then when you start thinking about it, then that's when you have to like really focus on moving your skeletal muscles to breathe. So in eunia, the diaphragm and external intercostal muscles are just contracting and relaxing on their own. It's just quiet breathing. Sometimes you take a very deep diaphragmatic breath where you're really focusing on contracting your diaphragm, this deep breath. So that's called diaphragmatic breathing. There's also the kind of the opposite isolation, costal breathing, which is just shallow breathing. You're only using those external intercostal muscles of your ribs. You're contracting and relaxing them, and so that's shallow breathing. So we have normal breathing or quiet breathing, which is diaphragm and intercostal. We have diaphragmatic, which is mostly diaphragm. We have costal, which is mostly just the intercostal muscles. And then kind of expanding beyond this, we have hyperpnea, which is forced breathing. And so that's going to require even more accessory muscles from places like your abdominal cavity. So when we're thinking about how to get quantitative about this, we might use something called spirometry, which produces a spirogram. So this is a pulmonary function test that you might use in a clinical setting. It's used to measure the volume and the flow of air. So when we look at this, we look at different types of volumes. The most important one is tidal volume. So this is the air that's just entering our lungs during quiet breathing. So you can kind of see those normal fluctuations in breath, so those smaller ones, and then the really high one and really low ones represent forced breathing. So when you go through this expiratory reserve volume, that's what you forcefully exhale past normal tidal expiration. So tidal is just kind of what's coming in and out of your lungs on just normal, eunia. When you push as much air as possible out of your lungs when you exhale, that's that expiratory reserve volume that's shown here in green. When you take in as much air as possible, that's the inspiratory reserve volume. It's the extra volume that can be brought into your lungs in forced inspiration, past that tidal inspiration volume. So we have in this spirogram a few not shallow breaths, but normal breaths. We have this breathing in, this inspiratory reserve volume, and then breathing out as much as possible, the expiratory reserve volume. And then anything else that's left in your lungs, this air that's left in your lungs after you exhale as much as possible, is the residual volume. And so that's going to be very important for preventing the alveoli from collapsing. And when we get different combinations of these volumes, we have different capacities. So for example, the inspiratory reserve volume and the tidal volume together make the inspiratory capacity. And then all of this together is the total lung capacity. So those capacities are different combinations or sums of specific types of volumes. So what that figure should also indicate to you is that not all air in the airway is used in gas exchange. We saw that some kind of sits around in your alveolus. There's also anatomical dead space, which is all the air in the respiratory system that doesn't even reach the alveoli. So it's just kind of being brought in and out, but not actually getting into those alveolar sacs. There's also that alveolar dead space, so that's air that's in non-functional alveoli. So maybe alveoli that are diseased, where they're not participating actively in gas exchange. And when you multiply these together, you get the total dead space. So that's all the air in the respiratory system that is not used in gas exchange. Okay, so this is the GIF that I thought we were almost to earlier. So this is a character from Adventure Time, breathing in and out. And if we get quantitative about this again, just like we did with the spirogram, we're thinking about respiratory rate. And so respiratory cycle per minute, or the breaths per minute in and out, is the respiratory rate. So this is important because it can be used as an indicator of disease state. Different conditions might change respiratory rate, and it also changes in response to different systemic factors, so things like oxygen and carbon dioxide levels, as well as pH levels. Remember that pH is really closely linked to carbon dioxide. If you think back, we talked about that briefly during digestive physiology, and that will keep coming into play throughout today's lecture. So just keep in mind carbon dioxide and pH are very connected to one another. Respiratory rate also changes as we age. So in an infant, the average respiratory rate is about 30 to 60 breaths per minute, so quite high, but then by the time an individual reaches adolescence, so ages 12 to 18, they usually decrease to about 12 to 16 breaths per minute at rest, which is fairly similar to what an adult respiratory rate is. So when we're thinking about how respiration is regulated, we talked about disease states, we talked about systemic factors. A lot of this ties back to respiratory centers in the brain. So these are located primarily in the medulla oblongata, or the medulla, and the pons. I'm going to briefly go over them and kind of state a little bit about what they control, but I'm not going to be testing you on these, just so you're not panicking about that. So in the medulla, there's the dorsal respiratory group, the DRG, the ventral respiratory group, the VRG, and so these control different types of breath, like forced breathing. And then there's the pontine respiratory group in the pons. The pneumotaxic portion actually inhibits the dorsal respiratory group, so it kind of interacts with some of the control from the medulla oblongata, allowing you to relax further after inspiration or after inhaling. And then there's the apneustic, which affects the depth of inspiration or the depth of that inhalation. So in addition to this kind of anatomical point, one thing to remember is that these respond to changes in systemic stimuli. So they respond to systemic stimuli, and we'll talk about that mechanism next. That's something that is important to know for the exam. So when we're thinking about kind of signaling and systemic changes, I mentioned that carbon dioxide is really important, and it is super crucial for regulating the rate of respiration. So one thing to keep in mind is kind of like our body is responding to carbon dioxide, how we breathe affects carbon dioxide, so it's a kind of feedback loop. And so when we hypoventilate, if you look at that word, hypo is like low, like hypothermic is low temperature, ventilate is breathing, so hypoventilation is holding our breaths, intentionally breathing very slowly, or breathing into a paper bag. And so when we do this, carbon dioxide starts accumulating in our systemic blood. So we see that here. When we hyperventilate, when we breathe too quickly, so breathing in and out super fast, carbon dioxide is removed from our systemic blood. So not only are we breathing oxygen in, we're more importantly almost breathing carbon dioxide out. Carbon dioxide is the important signaling molecule here. Something to note is that this is why sometimes swimmers will hyperventilate before they dive into the water, so then they can go longer before they have to take a breath. I know some of you are swimmers, so keep that in mind. Okay, so kind of tying together these ideas of carbon dioxide and pH levels and signaling, we'll start out with these central chemoreceptors. So what chemoreceptors Receptors actually are specialized cells. So the receptors are the cell part that sense concentrations of chemicals. So chemoreceptor, they're a specialized structure to sense chemical concentrations. So here, if we focus on these central chemoreceptors, that central part should tell you that these are in the brain and the brainstem. They're part of the central nervous system. And we see carbon dioxide being moved from the blood into the cerebrospinal fluid, or CSF. So here it's crossing the blood-brain barrier. Remember that there's tons of arteries that supply your brain, including the circle of Willis. So there's a lot of arterial blood kind of making its way in there systemically. And so this carbon dioxide is going to cross the blood-brain barrier, get into that CSF, that cerebrospinal fluid, and it's going to mix with water in that fluid to form carbonic acid. So that's what that H2CO3 is depicting there. This is catalyzed by an enzyme that we'll kind of talk about again later. And then that carbonic acid is going to dissociate into bicarbonate and protons. So hydrogen ions, those H+. Remember that a characteristic of an acid is dissociation into protons. And so this is making that cerebrospinal fluid more acidic, so lowering the pH. Remember, a low pH is acidic. And so we see that process here. And so that carbon dioxide is now going to indirectly increase the rate of respiration. So the way that it does that is that, we have this change in pH, and we have those central chemoreceptors right there. They are going to perceive that change in pH and send a signal to the respiratory centers in the brain in response to that pH change and higher proton concentration. So in response to that, the respiratory centers in the brain increase the rate and depth of respiration, and so that's going to remove carbon dioxide more effectively through the respiratory system and decrease levels of carbon dioxide in the blood. So it's a negative feedback loop. The opposite is also true. If you have low carbon dioxide levels, that's going to result in lower rates of respiration, so you're going to be accumulating carbon dioxide more slowly. So in addition to the central chemoreceptors, there are also peripheral chemoreceptors. And if we think about that word peripheral, that's kind of like thinking about the periphery, the outside, so not central, not in that central nervous system, but elsewhere in the body. And so these are found on systemic arteries, and the mechanism operates very similarly to those central chemoreceptors. So these are found on, indicated here, the aortic branch and the aortic arch and the carotid sinus. You can see that indicated here. The aortic arch bodies, or those aortic bodies, you can kind of see where the for peripheral chemoreceptors are, and then on the carotid sinus you can see that indicated as well. So these actually also sense dissolved oxygen. Remember that oxygen does not affect levels of the pH, but these peripheral chemoreceptors do sense changes in oxygen partial pressure. So when that partial pressure is less than 60 millimeters of mercury, they change and increase respiration rates. Keep in mind that the average in systemic arteries is about 70 to 100 millimeters of mercury. So it's important here that this happens in systemic arteries and that these chemoreceptors are located in those arteries. So why do you think it is important that they are located in those arteries rather than veins? Maybe take a moment and think about it. So remember, when we have systemic veins, those are returning deoxygenated blood from the body to the lungs. Even though it's not fully deoxygenated, we don't use a huge portion of the oxygen that's in our blood, but it's still quite lower, and so it's important that this kind of sensor and measurement take place in the arteries, because that's when a partial pressure of oxygen of less than 60 millimeters of mercury is going to really be out of the usual. So in addition to those respiratory centers, there are other parts of the brain that kind of tangentially affect the rate of respiration. In anatomy, you probably learned about the hypothalamus and the limbic system, and so those affect kind of different body responses. And if we have things like increased body temperature, feelings of excitement, so different emotions, the fight or flight response, or pain, all of these might serve to increase the rate of respiration. Something kind of interesting with pain, though, is that I've done lab experiments with students, so not in a research lab, but just kind of in a non-majors bio class, where students looked at pulse changes in response to pain. And in some people, pain increased pulse, and in other people, pain decreased pulse. And so there's a lot of evidence that pain kind of has different effects depending on different emotional health states. So for example, if you are very anxious, maybe snapping a rubber band on your wrist might help bring you more into the present moment and actually lower your pulse and lower your respiration rate and calm you down. So these are all very kind of nuanced and complicated, and there's a lot of factors at play there. So one thing that we can kind of look into here clinically is sleep apnea, which is a condition in which someone stops breathing while sleeping. This can range from seconds at a time to minutes at a time. And there's a couple of different types. There's obstructive sleep apnea, which is just kind of when those airways are blocked, and central sleep apnea. So obstructive sleep apnea is a structural blockage of the airway. This is due to a lot of conditions, kind of like obesity, but also other factors. And then there's also central sleep apnea, in which the respiratory centers don't have that response to rising carbon dioxide levels. In other cases, the diaphragm and the intercostal muscles don't contract regularly. And then if you have stroke or congestive heart failure, that can damage the brain and affect those respiratory centers. Medications such as morphine might also damage those centers. Okay, so moving on to our next section, I've used that term partial pressure a couple times with this P, and then when I've used it so far, it's been a lowercase p and then the symbol for the gas. But what that's referring to is the pressure of a single type of gas in a mixture of gases. So when we look at this, this is a diagram showing the partial pressures of oxygen and carbon dioxide in the alveolus of the lungs versus different systems of the body, the pulmonary artery and the pulmonary vein. And so we see these different concentration gradients for carbon dioxide and oxygen. So we know things are happening a little bit differently between the two. And so the gas that we breathe in, circulate through our body, and breathe out is a mixture of gases, not just oxygen and carbon dioxide, but we'll get to that in a minute. And when we talk about partial pressure, this is a measurement of the concentration of the individual gases in this mixture. So it's the pressure exerted from one particular gas within a mixture. We characterize this with Dalton's law, which states that the specific gases and mixtures have their own pressure, their own partial pressure, and total pressure is the sum of all of these partial pressures. So in this image here, we have three different chambers, one with just oxygen, one with just nitrogen, and one with a combination of those two. And so we can see that the pressure of oxygen is 159, the partial pressure of nitrogen is 593, and when we put that all together, the total pressure is 752 millimeters of mercury. So we see this summation of partial pressures. So atmospheric gas is a mixture. Even though we track primarily oxygen and carbon dioxide in our blood, and even though things like hemoglobin are more specifically designed to carry oxygen and carbon dioxide, when we breathe in, we don't control that. We don't say, okay, I only want to breathe in oxygen. We just breathe in whatever's in the air around us, with the exception of kind of, filtration through our nose hairs. But what the gas actually is primarily nitrogen. And so about 80% of the gas that we breathe in is nitrogen, but it turns out nitrogen doesn't dissolve super readily or stick around in our blood. So we don't really worry about that, with some exceptions that we'll talk about in just a moment. It's about 20% oxygen, and then much smaller fractions of it are water and carbon dioxide and other gases. So there's a lot of numbers on here. You don't need to focus on memorizing the numbers. Just focus on this idea that there's a lot of nitrogen in atmospheric air, a lot of oxygen, some water and carbon dioxide, and also focus on this 760 number, that the total atmospheric pressure is right around 760 millimeters of mercury at sea level. So one thing that I've mentioned before is that gases in the body move along their own partial pressure gradients. So again, this is a common theme of going from high to low. Substances move from high concentrations to low concentrations passively in a process called diffusion. Substances move from areas of high pressure to low pressure. We saw that when we saw the changes in the volume of the thoracic cavity that affected pressure and drew air in from the outside environment into the lungs. And then zooming way in and just looking at partial pressure of individual gases, gases in the body move from high partial pressures to low partial pressures. So this all really ties together. Partial pressure is very similar to concentration in a lot of ways, and often the higher the concentration, the higher the partial pressure, but there's some things that we adjust. So for example, with that nitrogen, it's very high in concentration in the air, but not high in concentration in terms of partial pressures within our bodies. So we'll kind of clarify that in just a moment. So in general, we say that those pressure gradients, those partial pressure gradients in the body drive the movement of gases between different systems, so between the blood and the lungs and between the blood and body tissue. So one thing to remember is that blood is a connective tissue, and when we look at this, we see that blood has plasma as its ground substance. Remember, it has cells, those leukocytes, thrombocytes, and erythrocytes that are kind of dispersed in this plasma matrix. And so because that ground substance is a fluid, blood behaves like a liquid, so we have to kind of study it like a liquid. And we're going to do that using something called Henry's law, which states that the concentration of gas in a liquid is directly proportional to the solubility and partial pressure of that gas. So we're going to kind of work our way through this formula. One thing that I kind of want you to keep in mind is that we're thinking about how gases enter a liquid, and we're also thinking about how gases exit a liquid. So let's say that we have a beaker, and that the beaker has a bunch of water in it. But regardless of whether the glass is half full or half empty, there's some combination of liquids and gases here. So we have this surface layer that separates out the liquids from the gas at the top, the headspace. And let's say that in this headspace, we have some of these green gases that are kind of higher in concentration, and some of these yellow gases that are lower in concentration. So when we're looking at these, they're kind of moving around, bumping around, and some of those green gases might make their way into the liquid. And some of them will kind of bump away from the liquid, but it follows that the higher the concentration or partial pressure of those gases, the more likely they're going to kind of enter into the liquid and cross the surface layer. Some of them might make their way in and then exit, but that exiting is also kind of based on certain properties like properties of the solute, the solvent, and the temperature. So when we're putting this all together, the concentration of gas that ultimately makes its way into a liquid is determined by the partial pressure, so how likely is the gas to get into the liquid in the first place, to enter it, divided by this constant, which is how likely the gas is to exit the liquid. So if we kind of take nitrogen as an example in atmospheric gas, the partial pressure of nitrogen in the atmosphere is extremely high. Remember, it's over 70% is that atmospheric nitrogen. But nitrogen doesn't like to stick around in blood, so it's also very likely to exit the blood. This K constant is quite high, and so these divide out and balance out, and ultimately the concentration of nitrogen in the blood is quite low. So if we think back to that decompression sickness case study that you might have read if you maybe looked at the week 1 slides, but that hopefully you remember from the first exam, decompression sickness is also called the BINS, and it has something to do with gas circulating in your cardiovascular system. So let's kind of think about this, about how it has to do with this range of atmospheric pressure and this idea that pressure is very high the deeper you get into the ocean compared to sea level. So there's 2 parts to this. There's the nitrogen narcosis part, and there's the BINS, which is decompression sickness. So as people are diving, they are going from an area of kind of normal atmospheric pressure, that's 760 millimeters of mercury, much deeper to higher levels of pressure. And so there's that part of it. But then also it turns out that the gas that scuba divers breathe in their tanks has a much higher partial pressure of nitrogen. So it's even more nitrogen than the atmospheric partial pressure of nitrogen. And so you have a lot of nitrogen entering the blood, which causes giddiness and symptoms similar to being drunk, basically. And so you have this part of it, the nitrogen narcosis. But then as you surface, as you go from deep in the ocean to back up to sea level, you're going from high pressure to lower pressure. If you do this slowly, then you have time to get all of that nitrogen out of your blood through your lungs. But if you rise too quickly, then nitrogen comes out of solution, it forms bubbles in your blood, and that causes joint pain, dizziness, shortness of breath, fatigue, paralysis, and unconsciousness. So those external factors affect how well nitrogen is staying in solution, and another way of stating that is it affects how likely the nitrogen is to leave solution, how likely the gas is to leave the solution. So nitrogen narcosis and decompression sickness are 2 parts of this formula. Nitrogen narcosis ties in with that partial pressure, how likely the gas is to enter the liquid in the first place. And the BINS, the decompression sickness, ties into how likely the gas is to exit the liquid. So if you want to treat and prevent the BINS, then you can go through a very slow ascent. So you can give your body time to get rid of that nitrogen through your lungs. You can also use a helium-oxygen gas mixture instead of a nitrogen-oxygen gas mixture while diving, or you can use this hyperbaric chamber, which is pictured at the right. This is a tank with very high pressure that's going to mimic the environment of the deep ocean. And so that is basically allowing you to go through this process of a slow ascent. So it puts you into an artificially high pressure environment and then slowly decreases that pressure, giving you time to remove the nitrogen from your body. It's also called a decompression tank or recompression chamber. So when you have gas exchange taking place, there's 2 different processes happening here, ventilation and perfusion. So ventilation is that movement of air into and out of the lungs. It's the airflow. Perfusion is the flow of blood into the pulmonary capillaries to actually receive the gases that are being brought in and taken out through ventilation. So the air has to get in, but it also has to make its way into the blood effectively. And so we see that pictured here. We have the alveolus in the lungs. We have the pulmonary artery and vein. It's implied that the pulmonary capillaries are in between. And we have the gas exchange taking place between the alveolus and the lung, or in the lung, and the pulmonary capillaries in the pulmonary circuit. So we see that pictured here. So if there's a gradient, if the oxygen gradient between the alveolus and the pulmonary capillaries is not strong enough, so if you don't have high oxygen or high enough oxygen relative in the alveolus relative to the capillaries, then the gas exchange will not take place. So that can occur if you don't have strong enough ventilation or healthy alveoli. So you resolve this by increasing ventilation, or you can redirect the flow of blood. So remember, we have sphincters inside of our blood vessels that can kind of help control going the flow of blood going from the arterioles to the capillaries. And so if we have an alveolus that's not effectively performing gas exchange, if there's a problem with that, then you change the flow of blood, redirect it to a more efficient area. So we have two processes happening here, external respiration and internal respiration. External respiration occurs in the pulmonary circuit in the lungs, with oxygen moving from the alveoli to those pulmonary capillaries. So again, there's a very strong concentration gradient here, or partial pressure gradient, I should say. The partial pressure of oxygen in the alveoli is about 104 millimeters of mercury. Remember that this is different from the total pressure. So we saw that the total pressure in the alveolus is about 760, but just of oxygen is 104. In the pulmonary capillaries, this deoxygenated blood has a partial pressure of about 40 millimeters of mercury. So we have a very strong concentration gradient. Oxygen is going to readily move from the alveoli to the blood and then be bound to hemoglobin. So it turns out that there is not a lot of oxygen that's actually dissolved into our blood. Most of it is bound to hemoglobin. We'll talk about different binding conditions in a little bit. The lungs are really important for removing carbon dioxide as well. So when you inhale, the goal is to bring in oxygen, but when you exhale, it's very important to get rid of carbon dioxide, which is an important waste product from cellular respiration and also an important regulatory component, as we saw earlier. So the partial pressure of carbon dioxide in the alveoli is 40 millimeters of mercury. In the pulmonary capillaries, it's 45. So again, it's going from an area of high to low, going from the capillaries to the alveoli. And even though there's not a really strong concentration gradient here, carbon dioxide tends to be much more soluble in fluid in both the blood and the alveolar fluids. So it's able to still effectively move in a rate that is helpful to us. So that was external respiration. When we're thinking about gas exchange in terms of internal respiration, that's occurring in body tissues in the systemic circuit. So this is occurring along an opposing partial pressure gradient. And so this is happening kind of the opposite of the flow of gases in the pulmonary circuit. Now we have oxygen going from blood into body tissues and carbon dioxide going from body tissues into blood. So you should know kind of the flow of gases, of oxygen and carbon dioxide, how it's different for external and internal respiration. One thing that I think is kind of interesting to keep in mind that I don't think I've talked to you guys about, though maybe I have, but a lot of people think that deoxygenated blood is blue and oxygenated blood is red. That's not true. Oxygenated and deoxygenated blood are both red due to properties of hemoglobin, but they look slightly different. So oxyhemoglobin is this bright red color. Deoxyhemoglobin is kind of a darker color, and when we look at our veins, our systemic veins, which carry deoxygenated blood through our skin, it looks blue, but that's just because of how light refracts and the properties of this deoxyhemoglobin, which is still red. So when we look at the structure of hemoglobin, we can kind of notice a few things. One is that hemoglobin has 4 different kind of subunits to it. It has 4 chains of the globin protein. Each of those has kind of repeating structures that we'll talk about in just a moment. So you see those 4 different chains. Each of those is going to be able to bind to oxygen. And so heme is found at the center of each of those chains, and that is a red pigment with an iron ion. That's what that Fe is in the center. So that iron ion at the center of heme is going to be important for oxygen binding. And because there's 4 different subunits, 4 oxygen molecules can bind to each hemoglobin molecule. And it turns out each red blood cell has about 300 million hemoglobin, and so that means that each red blood cell can carry 1.2 billion oxygen molecules. So just a ton of oxygen being carried by this hemoglobin. But we can get a little bit more quantitative than that. So when we're thinking about how this binding occurs, you might think back to kind of intro bio, think back to enzymes, and remember that when a substrate binds to an enzyme or binds to a protein, there's a conformational change. And so here, binding the oxygen changes the structure of the hemoglobin, and it kind of happens along a sequential level. So oxygen binds to the first subunit, that causes a conformational change. The oxygen the second oxygen is then able to bind to the next subunit a little bit more quickly. So as we have these conformational changes, the oxygen is going to be more and more efficient in how it binds to the hemoglobin. So when we have this hemoglobin carrying 4 oxygen molecules, we say that it is fully saturated, and that's what we're going to be measuring. So when we calculate this out, it turns out that the percent of hemoglobin that are saturated in a healthy adult is anywhere from 95 to 99% of the hemoglobin. And when we plot this, so if we're plotting oxygen saturation as a function of partial pressure of oxygen, we get what's called an oxygen hemoglobin dissociation curve. So this is describing that relationship of partial pressure of oxygen to the binding of oxygen to heme, and then also its dissociation from heme. So that straight part of the curve here is indicating the range of partial pressure and oxygen saturation in the systemic capillaries. We see that really tiny changes in partial pressure here is going to lead to huge changes in oxygen saturation, but in the lungs this kind of plateaus out, and the flat part of the curve indicates the range of partial pressure in the pulmonary capillaries. At that point, changes in partial pressure aren't going to dramatically impact the oxygen saturation of hemoglobin. So there's a lot of factors that kind of shift this curve around. And so here we can see that if you have increased affinities for oxygen, that might be due to decreases in the partial pressure of carbon dioxide, decreases in pH, or sorry, not decreases in pH, increases in pH and decreases in hydrogen ion concentration. This thing called 2,3-DPG, which I'll talk about in just a moment, and decreases in temperature. The right shift, a decreased affinity for oxygen, might be due to increased carbon dioxide and increased proton concentration, so decreased pH. Increases in this 2,3-DPG and increases in temperature. And it turns out that the relationship between pH and hemoglobin affinity for oxygen is called the Bohr effect. So if you see that Bohr effect, it's referring to that relationship between pH and oxygen binding to hemoglobin. So what that 2,3-DPG actually is kind of a placeholder. So it fits into deoxyhemoglobin, hemoglobin without the oxygen, to maintain its shape. And it's actually really important for directing oxygen from a maternal source to the fetus. So remember that the circulatory systems of a fetus and the parent are kind of linked in certain ways, specifically through the placenta. One thing that's kind of tricky though is that there's not a very strong partial pressure gradient, so it's hard for oxygen and CO2 to flow effectively, but fetal hemoglobin has a stronger binding affinity for oxygen than adult hemoglobin does, so it's able to really effectively grab that oxygen. So I've mentioned that carbon dioxide does things a little bit differently than oxygen. It's not primarily, or it is, it's not primarily carried by hemoglobin. About 10% of it is bound to plasma proteins, including hemoglobin, but 5 to 7% of it dissolves pretty readily into blood plasma, which is way more than oxygen does. But then a huge portion of it, 85%, is carried as part of this bicarbonate buffer system. So this is something that we discussed a little bit in digestive physiology when we were talking about parietal cells. At that time, I didn't ask you to really understand it thoroughly, but I definitely want you to understand the mechanism a little bit more this time around. So here we have those parietal cells pictured. On the left, we see the lumen of the stomach, so the inside of the stomach where there's a lot of hydrochloric acid. On the outside, we have the interstitial fluid and capillaries. So earlier I mentioned that the relationship between carbon dioxide and water transitioning to carbonic acid is catalyzed by an enzyme, and that enzyme is carbonic anhydrase. So that's going to form that carbonic acid from water and carbon dioxide. That is then going to dissociate, because it's dissolved in this aqueous environment, into bicarbonate ion and a proton. So you have this buildup of bicarbonate and of protons. And the bicarbonate is very important because that causes what's called a chloride shift. So you have this bicarbonate accumulation, and it's going to actually go through these channels and be exchanged with a chloride ion. So there's movement of bicarbonate in exchange for chloride. Then you have hydrochloric acid. You have the H plus and the Cl minus ions moving around, which is really important for the stomach. But this bicarbonate buffer system is important for the whole of your body. And it's a major way that carbon dioxide is carried through the body. So that kind of occurs in reverse when you're in the pulmonary circuit and releasing carbon dioxide. So going from bicarbonate and hydrogen ion to carbonic acid and then back to water and carbon dioxide. So carbon dioxide does also bind to hemoglobin, but it doesn't bind in the same sites. It doesn't bind on that heme part of it. It actually sticks to these amino acid groups or moieties that are on the globin portions of the hemoglobin. That forms carbaminohemoglobin, which is a fancy way of saying carbon dioxide attached to aminos on hemoglobin. So we saw this slide earlier about the different types of breathing involving different muscles. Let's focus first on this hyperpnea, or focus again on this hyperpnea, which was that forced breathing where you're using a bunch of accessory muscles. So when you have hyperpnea, you have this increased depth and rate of ventilation, and it's meeting a demand. So you might have an increase in oxygen demand due to exercise or a disease state. And because you are trying to meet a need that you actually have, the partial pressures of oxygen and carbon dioxide in the blood, as well as blood chemistry, don't fundamentally change. So everything's kind of balanced out. It's part of this system. But when you hyperventilate, that is kind of more forced, throwing the system kind of off. And so it does change the partial pressure of carbon dioxide, and as a result of blood chemistry, because it's not meeting a need. It's kind of a different state that's abnormal, and so you're decreasing, for example, with hyperventilation, you're decreasing the amount of carbon dioxide, the partial pressure of carbon dioxide, because you're getting rid of more of it. That's increasing pH and becoming more alkaline. So one other kind of different condition to keep in mind, we talked about decompression sickness, about the bends, which represents going to an area that's much higher pressure than atmospheric pressure. But if you climb a mountain, you decrease atmospheric pressure, you lower the partial pressure of oxygen, and that can lead to a lot of problems as well. So there's a lot of kind of ways that we get around these problems, these compensatory mechanisms. So one is that oxygen is more readily released from our tissues at higher altitudes, so are released into our tissues. So hemoglobin is not hanging on to that oxygen as readily. It's more able to kind of release it and provide it to our tissues. And that molecule I mentioned, that 2,3-DPG, that also increases at higher altitudes, meaning that, again, oxygen is more easily dissociated from hemoglobin. You can also behaviorally go through acclimatization, where you slowly ascend a mountain. So you kind of move very slowly, and then your kidneys are going to upregulate EPO, which is erythropoietin, which is going to signal your body to produce more red blood cells. So even though you have changes in the hemoglobin, even though you have changes in that atmospheric pressure and the hemoglobin isn't going to be fully saturated, you have more red blood cells to make up for it. So if you don't do this, you get what's called acute mountain sickness, or AMS, also called altitude sickness. And so in this, you have this condition from high altitudes that causes nausea, vomiting, fatigue, lightheadedness, drowsiness, feelings of disorientation, increased pulse and nosebleeds, and may sometimes cause pulmonary or cerebral edema. So I know on the last exam, a lot of you maybe were not too sure about that question about different requirements of life and the bends. So hopefully because of this lecture, you see that a narrow atmospheric pressure range is a very important prerequisite for life, and that if you lower it too much, as is the case with altitude sickness, or you increase it too much, like with the bends, that can cause a lot of serious ramifications for your respiratory and your cardiovascular systems. So this was the respiratory physiology lecture. The new lecture for this week will be on blood and lymph, so specifically looking at those systems, so kind of what's flowing through these systems that we've talked about other than gases. So I will get that posted in the next couple of days, and then you will have a study guide posted when the next quiz comes up. I am still going to post that exam review resource from lecture exam 1. I'll send out a quick message when that's posted. So that will be to prepare you for the final exam, which is semi-cumulative. Please don't hesitate to email me if you have any questions in between reflections. Other than that, I hope this made sense, and good luck.